Hello everyone. The topic of today's lecture is two-dimensional gel electrophoresis. And two-dimensional gel electrophoresis is a powerful technique in proteomics in order to resolve thousands of proteins. We know that in proteomics workflow, in proteomics we study entire proteome of a cell. Entire proteome means there are 10,000 of proteins which are being present in a cell, and we study them simultaneously. And it is not possible to resolve thousands of protein using single property of the protein for separation. That's why two-dimensional gel electrophoresis, it's a very powerful technique which is employed to study thousands of protein to separate them. In fact, the entire proteome is separated using 2D GE. And in the workflow, there are two steps. First is separation. All the proteins in the proteome, they are separated. And after separation, the proteins are identified using mass spectroscopy based methods. So the very first step in proteomics is extraction of the proteome. The entire proteome, the thousands of proteins, they are extracted from the cell and they are run in two-dimensional gel electrophoresis in order to separate them. In this lecture, we'll discuss about the separation step in proteomics, and that is composed of two-dimensional gel electrophoresis in gel-based proteomics approaches. Two-dimensional gel electrophoresis or 2D page, it separates entire proteome on the basis, in first dimension on the basis of their isoelectric point, and in second dimension on the basis of the size of the proteins. And that's how, because it uses two different properties of the proteins to separate them, 2D GE is capable of separating thousands of the proteins simultaneously. First, proteins in the proteome, the entire proteome is separated on the basis of their pI, and that is called isoelectric focusing. So isoelectric point of a protein is the pH where the net charge on the protein is 0, because at that pH the total number of positive and negative charge would be equal on the protein. And the pI of a protein obviously depends on the composition of the protein in terms of amino acids. A protein is having more number of negatively charged acidic amino acid would have lower pI. It's called acidic protein. In contrast, a protein having large number of more number of basic amino acids would have a basic pI, the high value of the pI, and it's called a basic protein. And pI of a protein can be determined experimentally by a technique called isoelectric focusing. In isoelectric focusing, a pH gradient is made in a tube, and this is a pH gradient. For example, here this pH 3 to pH 9, and protein sample is loaded in the pH gradient, and it's loaded at negative electrode here. So sample is loaded at the negative electrode and at very high pH, because at very high pH all the proteome is composed of all type of proteins, acidic, basic, neutral. So when you load the proteome at high pH, all the proteins present, irrespective of their natural charge, will acquire net negative charge, so that all the protein can move from negative electrodes to positive electrode. And as a result, when the protein migrates across the pH gradient, whenever a protein reaches to the range of pH which is equal to its pI, it will have net charge 0, and hence it will not move under electrical field, and it will stop there. So the gel can be stained, and we can see different bands at different pH range which are separated on the basis of their pI values. And in a typical experiment, like you have pH gradient like this one, this is positive electrode and this is negative electrode, and the proteins are loaded at high pH on negative electrodes, so all the protein would have net negative charge. If you are loading protein at a positive electrode, in order to make them migrate towards negative, you require to keep at very low pH, so protein would have net positive charge. But here Generally proteins are loaded at negative electrode and very high pH, so all the protein can move irrespective of their natural charge toward the positive electrode. So protein will start moving when you attach the electrical field, because all the proteins are negatively charged, so they will move across the pH gradient, and across the gel the high molecular weight protein will move naturally slow in comparison to the small molecular weight proteins. But because IEF is run for a longer period of time, so slowly the proteins which are having similar pi, or the same molecules having same pi, they will catch up, and eventually they are separated on the basis of not size but their respective pi values. So you will have more acidic proteins towards positive electrode and more basic proteins toward negative electrode. And this pH gradient in isoelectric focusing is generally made using a mixture of carrier ampholytes in a soft acrylamide tubes. But in today's time you have replacement of the carrier ampholytes in making pH gradient in isoelectric focusing, and that is by immobilized pH gradient strips. IPG strips have many advantages over carrier ampholytes. That's why they are in high uses in 2DG in these days. In IPG strips you have buffering acrylamide derivatives containing free carboxyl groups and tertiary amino groups in order to generate a pH gradient, and the length is around 107 millimeter or 176 millimeter. That's also customizable. And you have a sample cup where sample is loaded, that's on the negative electrode, and this gel in the tube, this contain a pH gradient of a definite pH value. That's also customized depending on the requirement. You can have a pH gradient say 3 to 10 pH, or you can have 3 to 7 pH. As per requirement it can be made. And the pH width is 0.1 to 1.0 here. You can also choose what kind of range of pH you require in making pH gradient in IPG strips. They have many advantages over carrier ampholytes, like they give better resolution in 2DG. They have unlimited stability of the pH gradient. In carrier ampholyte, if you run isoelectric focusing for longer period of time, eventually there is a disruption of pH gradient, and that does not happen in case of IPG strip. In IPG strips, you have flexibility in the choice of pH interval. You can choose as per your requirement. You have high loading capacity. You can load very high amount of sample in IPG strip. Their reproducibility is also high. And in case of carrier ampholytes, when you run them for longer period of time, due to electroosmosis, solvent flows towards cathode and eventually leading to disruption of the entire pH gradient. That is called cathodic drift. So cathodic drift is not there in case of IPG strips, and you can have full control of pH, buffering capacity and ionic strength when you are using IPG strips. And that's why IPG strips are, in today's time, they are very commonly used in running isoelectric focusing in 2DG. So once you have run proteins on the basis of their pi in isoelectric focusing, that IPG strip can be taken off. It can be treated with the sample buffer, the lamellae buffer, which is used in SDS page, where you have reduction and alkylation process. You reduce disulfide bond, alkylate cysteines, and then this strip, IPG strip, can be loaded on the top of a precast SDS page. And this SDS page, you allow it to run in perpendicular direction to the isoelectric focusing. So proteins would migrate now in perpendicular direction from IPG strip, and eventually all the proteins in the proteome would be separated in this second dimension, which is SDS page. Here migration will be on the basis of size of the protein, and once the gel is completed, the run is complete, the gel can be stained by different dyes like Comessi blue or silver staining can be done. In fact, fluorescent dyes, they are very sensitive, which are also commonly used to detect even a small amount of the protein in 2DG. And once you stain the gel, you will see many spots like this. And here on x-axis you have 3 to 10 pH range. This is the pH range which you have chosen in IPG strip. So this is 3 to 10, and in perpendicular direction you have proteins have migrated on the basis of their size, so you have molecular weight also. And each spot can be given a specific pi value, and which is coming from here x-axis, and this molecular weight from the y-axis. And most of the proteins, they are resolved in the second dimension of 2D GE, and only proteins which cannot be resolved in 2D GE are those which have very nearby pi value and very close by molecular weight, and that is rare in case of proteome. That's why in 2D GE most of the proteins of the proteome, they are resolved well. And SDS has a special role in SDS page. SDS is a negatively charged anionic detergent. So it gives negative charge to the protein. It binds extensively to the protein molecule. In fact, 1.4 gram SDS binds per gram of the protein. And that's why by extensive binding of SDS cause disruption of a secondary structural element in the protein, and protein become denatured, protein become completely unfolded. And as a result, the native charge on the protein is masked. Irrespective of the native charge present on the protein will migrate, will have net negative charge, so it will migrate towards positive electrode in SDS page. And because SDS bind extensively, it also prevent aggregation of the proteins, and same time it's also disrupt if there are any non-covalent intermolecular interactions. And most importantly, it also, because denature proteins completely, the shape of the protein also disappears. So any difference in migration arising because of a difference in shape of the protein, that vanishes here. So protein exclusively migrate on the basis of their size. Shape has no role to play because shape is disappeared due to denaturation in presence of SDS. So the gel is complete two-dimensional gel. So what do you see here? You see a pH gradient on x-axis, on y-axis you see molecular weight, and what you can infer from here, you can infer total number of protein. These spots, each spot correspond to one protein. So these spots can be counted, and total number of protein present in the sample in the proteome, those can be found out from here. And relative intensity of the spots, because you see some spots are very thick like this one. So the intensity depend on abundance of the protein. A protein present in high concentration naturally will give a very thick spot. So this spot is overexpressed. This protein is in high concentration. That information we can derive from here. And we also can derive information about differentially expressed proteins. We can compare gel from two different conditions, and we can find out which are the proteins being expressed differentially in either of the sample. And we also can record change in expression profile in terms of change in environment or physiological conditions. So for example, if you want to see proteins being expressed in stress condition in comparison to normal, 2DG can be run for the normal sample and the stress sample, and both the gel can be compared in order to see what are the proteomic profiling changes occurring due to stress. And each protein, each spot can be given molecular weight and the pi value. So once proteins are resolved, the second step in proteomic study is identification of the proteins. So each spots can be identified using different techniques. For example, Western blotting is one of the method to identify spots. If you want to know a particular protein present in the gel or not, you can use antibodies against that protein, and using Western blot method can be identified. Or Edmond degradation, this spot can be excised. Any spot which you are interested to identify, all of them individually, or some spot if you are interested into, that can be excised from the gel. After cutting from the gel, it can be identified. And Edmond degradation is a chemical sequencing method. This also we have discussed earlier, that can be employed here in order to identify a protein. Or peptide mass fingerprinting and tandem mass spectroscopy. These are two very common methods which are based on mass spectroscopy and which are used in proteomic studies for identification of proteins. So these we'll discuss in next separate lecture. So, and this 2DG also can be used in differential expression proteomics. For example, you have a cell, a control cell and a drug-treated cell, and you want to see which are the protein present in drug-treated cell, or which genes have increased expression because of presence of the drug. So you can run 2DG from both the samples separately. You can extract protein from individual sample from the control cell as well as drug treated cell. You use identical condition to run isoelectric focusing, then reduction and alkylation to run SDS page in the second dimension. And once SDS page is run, you visualize the protein using silver stain or any other stain. And after that you have got two gels, one gel from control cell and another gel from the drug treated cell. Now these two gel can be superimposed on each other to see difference in expression or difference in presence of the protein spots. For example, here you see that there are two spots which are present only in drug treated cell. They are marked with two arrows here. Now you would like to identify them in order to see which are these two proteins being present in drug treated cell alone, not present in the normal conditions. So these spots can be excised from here, and these can be sent for in gel digestion separately. And finally they can be identified using mass spectroscopy, peptide mass fingerprinting or MSMS, tandem mass spectroscopy methods. So eventually you will know that these are the proteins which are being present in the drug treated cell which are not present in the control cell. So this is called differential expression proteomics. Now 2DG is extensively used in proteomic studies, but it has limitations also in terms of resolution, because you have thousands of proteins present, and the resolution is a major problem. Resolution means how distinct the spots are. When the proteins are too large in number, they are too nearby, that leads to poor resolution. Then sensitivity, because many proteins are super abundant in the proteomic sample, they are present in very high concentration, others are very rare. So to visualize the superabundant protein separately and to enrich the scarce protein which are present in rare low concentration is also challenging in 2DGE. And then representation, not all protein present in the cell are represented in same condition. For example, there are proteins which require special treatment for solubilization, like membrane proteins, or there are proteins which are having very high PI. So to solubilize them, to make them run in 2DG, you require changes in buffer conditions. Then automation is another issue, because you run gel, you visualize them yourself, eventually you pick the spot in order to identify. So automation is also required. Manual interference causes problem in cutting the spot and identifying them. Resolutions in 2DG can be improved in several ways. For example, a length of IPG strip. You look at this gel here, there are so many crowding, so much crowding is there, so many spots are nearby. So that leads to very poor resolution. It's very difficult from here to cut individual spot to identify them. If you cut a spot, they'll be mixing from neighborhood spots also, and that's why you cannot identify in a poorly resolved gel. So you need to improve the resolution. So one way to improve resolution, that means distinctness, separateness of the spots, how separated the spots are, that's defined in terms of resolution. So length of IPG strip can be increased. It's been seen generally more than 30 centimeter long IPG strips that give a proper resolution of a proteomic sample. Size of the gel can be increased, but there is a limitation. You cannot keep on increasing size of the gel also, but Nevertheless, if you increase size or you take a bigger gel, resolution become better. Flattening of the pH gradient. If you see most of the protein, they lie in a particular range of pH. That pH range can be flattened in order to improve the resolution. And zoom gels is another strategy which is used to improve resolution, where multiple range isoelectric focusing can be run with narrow pH ranges. For example, you are running a 3 to 12 pH range and it's very crowding, resolution is poor. You can run 3 to 7 IEF in one sample, and second time can run 7 to 12. And these two gel, you can put them together for final analysis of the proteome. So that's called zoom gels. Pre-fractionation studies also, they are adopted to improve resolution. You can remove some of the protein or you can remove half of the protein, run them separately using chromatographic techniques. And sample concentration also can be changed. For example, if all the spots are very dark, it means sample is very high. That's why they are closing nearby to each other. So you can reduce the sample concentration in order to enhance resolution. So flattening of the pH gradient. For example, we see here most of the protein, they lie between 5 to 6 pH range. So 5 to 6 pH range can be flattened here. If you see in the IPG strip, this is from 4 to 7. And most of the protein are 5 to 6. So you can run from 4 to 5 and 5 to 5.5 and 5.5 to 6. So you can flatten the range where most of the protein lies, and they are creating crowding to compromise resolution. Or you can try zoom 2D gels, where for example this is 3 to 12 pH range, and we see that there is a lot of crowding. The resolution is very poor here, and it can be run like for example 4 to 7. If you run 4 to 7 pH here on the right hand side, you see these spots are all well resolved. So you can improve resolution by using zoom gel. So you can run 4 to 7. In the next gel you can run 7 to 10 or so on, and eventually you put them together to see the entire proteome. Then pre-fractionation studies are also being used. For example, if you have a protein which is present in very high concentration, like in case of human fluids. In human fluids, albumin is one of the very common protein found in very high concentration, and you have very big spot for albumin. It's so thick that it covers the neighborhood spots also, and that's compromising the resolution. So what one can do, you can remove albumin before running 2D GE. You can use a column, affinity column. In affinity column you have anti-albumin antibodies, and you run your sample on this column. So the albumin is retained by the affinity matrix, and non-albumin protein, they are coming out of the column. Those can be run in 2D GE. That's called affinity depletion. So you can deplete superabundant protein from the gel, from the sample before running 2D GE in order to enhance resolution. Or you can pre-fractionate. For example, you can run ion exchange before going to 2D GE. You can separate acidic protein and basic protein, and acidic protein can run separately on 2D GE, basic protein can run separately, and then eventually both the gels can be seen together. That will also improve resolution. Or another way is affinity enrichment. For example, if you want to do glycoproteomics, you just want to see the glycoprotein in the proteome. You are not interested in entire proteome, or you just want to see phosphoprotein. You want to do the phosphoproteomics. So you can remove glycoproteins or phosphoproteome as per your need, and you can run only those protein where you have interest, rather than running entire proteome, and that also will enhance the resolution. So that is done by using affinity chromatography. That's called affinity enrichment, where you have the stationary phase of the chromatography column is made up of beads coated with lectin. Lectin is a protein which binds to glycoproteins. So you have a lectin immobilized in the stationary phase of this affinity column. So when you load the sample on the top, so all the glycoprotein will bind to the matrix, and the non-binding protein will be eluted out unbound. Then those bound protein, they are eluted using sugar concentration. So you have separated lectin binding protein, which are essentially glycoproteins, and non-binding protein which you are not interested into. So you can run the lectin binding protein in 2DG to perform the glycoproteomics. Or similarly, if you are interested in phosphoproteomics, in the affinity column you can use antibodies against serine phosphate, threonine phosphate or tyrosine phosphate. We know that in proteins phosphorylation occurs on the side chains of serine, threonine and tyrosine, and antibodies against those serine phosphate, tyrosine phosphate, they are available. So you can immobilize antibodies and you can separate phosphated proteins, and those phosphated protein can be run in 2DGE. Or in fact, instead of antibodies, you can use metal affinity chromatography, aluminium based metal affinity chromatography, in order to enrich only phosphoproteins from the proteome, and then can be studied in 2DG for proteomic analysis. Another issue is sensitivity. In proteome, there are thousands of proteins and they all vary in their concentration also. The proteins in a cell differ in abundance over 4 to 6 orders of magnitude. Some proteins are super abundant and other proteins are rare. They are in very limited concentration, very less concentration, and you need to see all of them together. That's a challenging task. And the sensitivity problem, because you cannot have a single dye which can show simultaneously superabundant protein which are in very high concentration and the protein which are in very little concentration. So in that case, there are two type of problems based on sensitivity comes in case of 2DGE. The first is the difficulty in detecting the rarest protein at all, the protein which are rare. You cannot detect them along with the superabundant protein. So for that, what one can do, one can enrich the rare protein separately and then run 2DG. So you have to change the stages depending on your need, depending on your objectives. The second problem based on sensitivity is tendency of the spots produced by abundant protein. The most abundant protein, it will create a very big spot and it will mask other proteins which are in vicinity, and that creates problem. This problem can be taken care of by affinity depletion, which we have just talked. So the use of narrow range IPG gel in combination with prefractionation or affinity depletion of very abundant protein, that is applied to tackle the sensitivity problems in 2DGE. Then another issue in 2DG is representation. You are running thousands of proteins here, entire proteome, and all the protein, they have a great diversity in case of their chemical and physical natures. So it's not possible to have a single buffer to solubilize them, and that's why all the proteins present in the cell, in the proteome, they are not represented by 2DGE. You need to change a different strategy to look at the different type of proteins, and the major problems comes at solubilization steps. In general standard lysis buffer in 2DGE, that uses a chaotropic agent like urea, and along with the urea you have non-ionic detergent such as NP40. You don't use SDS here, because if you use SDS before running 2DG, that give negative charge to all the protein and you cannot run isoelectric focusing. So you use here non-ionic detergent like NP40, and along with that you have a reducing agent which can break disulfide bonds. And this standard lysis buffer is not applicable in case of membrane proteins, and also for the proteins which are having very high PI, for example histone proteins or chromatin proteins. So in case of membrane protein, you need to use a detergent, a stronger detergent, non-ionic detergent like CHAPS, so it can solubilize membrane proteins. And in fact, to deal with membrane protein, one can separate the membrane, entire membrane separately and extract the protein individually. So the membrane protein can be separated in comparison to the cellular proteins. And histones proteins and ribosomal protein, they have very high PI value, they are highly basic. For them, acid urea gel and acid urea tritone gel, they are specifically used to visualize them in 2DGE. Then because in 2DG you need to run 2DG and you need to visualize the spots which are very nearby also. So there you require automation because isolation of every spot is important, and then after isolation one has to cut the spot in order to identify. So manually it's very challenging, so you require automation. In fact, there are robotics available which can excise spots. For example, here these spots, these can be excised by the robotic system. It can be picked, and these spots can be, after excising from the gel, can be transferred to the multi-well ELISA reader plate for further processing in order to identify them. So that was gel-based proteomics. Despite of all these limitations, 2D gel is a very powerful technique to resolve proteomes in gel-based proteomics. But without running 2DG also you can do proteomic studies. You can resolve thousands of protein, and those are part of non-gel based proteomics, where you can use multi-dimensional chromatography. One can use isotope coded affinity tags, or you can label proteins using different tags. You can label protein in culture media, that's in CELAC, or you can use protein arrays. So these non-gel based proteomics approach will discuss in a separate lecture.